Our next speaker is Iman Hajirasouliha from Cornell. Iman is an associate professor in computational biology, and he brings to us a unique perspective. Moving forward in our field, we'll be looking to bioscientists, biostatisticians, and mathematicians to influence our interpretation of these tools. And I think Iman brings a unique perspective. He's extensively published in this area, and we'll look forward to his comments regarding the tools that we have and how to critically look at each of these. Iman, thanks so much for participating. Look forward to your comments.
Hi, everyone. Thank you for the introduction. This is Iman Hajirasouliha. I'm from Weill Cornell Medicine, and today I'm going to talk about precision medicine and AI, especially in applications to IVF. So just a quick background of my research lab. My lab is a computational lab at Weill Cornell Medicine, and we are focused mostly on genome sequence analysis and biology, especially working on looking at human genome characterization. And we are also doing a lot of deep learning image classification work, especially with applications of AI and deep learning in IVF. So just a few years ago, there was this article in the Wall Street Journal saying, IVF often doesn't work. Could an algorithm help? And since then, there's work by my group and many others to sort of showcase AI and machine learning can be useful in the field of IVF. So essentially, we want to see when AI meets IVF, can we improve the chance of live births in IVF processes and essentially make healthier babies for future. So some quick background on IVF. We know that many women in the US and around the world have problem conceiving, around 10% of the population actually. And more people are turning into assistive reproductive technologies such as IVF to increase their chance of pregnancies. IVF has a high cost. So on average, this varies among centers, obviously, and among different countries, they have different cost systems. But usually it's about $10,000 to $15,000 per cycle. So it's a huge cost for average families. And the success rate is still pretty low. So often less than 50%, especially for older patients. The IVF success rate is significantly lower. And there are also other major concerns. For example, because the success rate is low, often you would transfer more than one embryo, and this would lead to a higher chance of multiple pregnancies. And also it would essentially add to more complications for mother and the child. So another important factor here is the concept of aneuploid versus euploid embryos, especially for, again, older patients. Many of the embryos become aneuploid, meaning there are, for example, extra chromosomes or some rearrangements in chromosomes that can be lethal. So aneuploidy also often results in significant consequences, including miscarriage and birth defects. So it's also important to identify aneuploid embryos and as much as possible avoid transferring those embryos. So as I mentioned, there are several factors contributing to a successful outcome for pregnancies. And one important factor is the selection and transfer of euploid embryos. So you want to select embryos that have a higher chance of live birth and with less complications and for the most part, a single healthy baby. So AI can be helpful, and this is like sort of like a question we are asking and many others are exploring in recent years. So can AI make our life better? And this is, there are several questions. One, like I'm giving two examples in the presentation, one for assessing embryo morphology and one for assessing embryo aneuploidy. So as I said, these two questions are one of the like basically important questions in this field. There are many other questions that you can ask. For example, you can check the quality of sperms, or many other, you know, like automatic, many other, you know, like classification or, you know, like assessments in the entire IVF process. But I'm just giving you like these two examples in this presentation. And these are two of the questions that our group actually worked on recently. So historically, morphology assessment was done manually in embryology labs, for example. And often embryologists looked at the specific features of the embryo images. For example, there are different grading systems. Embryology would look at, you know, the inner cell mass or the, you know, teropectoderm, expansions of the embryos. And essentially they would give, you know, like different grades based on these features. This is usually non-invasive because, you know, like embryology would only look at the images of embryos, and it's relatively inexpensive. However, it's not a standard, meaning there is heterogeneity and there are, you know, like intra and inter-observer bias. So different embryologists may have different opinion, and it can be also time consuming because especially for some of the most difficult cases, their embryologists spend a lot of time assessing these embryos. There are other technologies, for example, the time-lapse microscopy technology that would, you know, like give you images of embryos during like different time points. For example, one machine that is being used at Weill Cornell and at many other clinics, you know, like look at the development of embryo from day 0 to day 5 or even like 6 or 7. And every, say, 20 minutes, you would get an image of the embryo. So you can have essentially several hundred frames, and you can look at these embryos, like the development of the embryos, and see the time points and, you know, like the quality of the embryos at each time. So again, this is a non-invasive procedure, and you have more images to assess. When embryologists look at these images, but these machines are costly and not widely used. So again, you know, like there is this trade-off because, you know, like not everyone can afford these machines. So many labs only look at like single images for assessing embryos. For detecting aneuploid embryos, the most common way now is the PGTA testing. This is done invasively, meaning you have to do a biopsy. So you would take out a few cells from outside the embryo, you sequence the DNA. So it's a pretty expensive procedure. You have to send samples to a lab. And occasionally you get, you know, false positive and false negative due to the mosaicism nature of embryos, because not every cell harbors the same aneuploid or euploid features. So the main question that we asked is if an AI trained on thousands of embryos that we had before, without any human intervention, can reliably predict embryo quality and and also assess the ploidy of embryo. So it can also distinguish aneuploid versus euploid embryos. And this would increase successful pregnancy rates, reduce complications, and makes this procedure a fully automated approach. So you don't need human intervention. So everything would become standard using algorithms. So my group, a few years ago, wrote one of the first papers in this domain. We published in Nature Digital Medicine, led by former postdoc Pega. And the idea was to train a deep learning model using thousands of embryos and labels coming from a panel of expert embryologists. So we have several embryologists. We use the consensus labels from this panel to grade embryos and train a neural network that uses these labels to predict. This is mainly basically a classification task now, to predict good versus poor embryos. So previously, there were some machine learning approaches, and people are still like using some machine learning approaches, mostly, to focus on specific features of the embryos. But the deep learning approach would let us look at the whole picture at the same time and get, like a holistic view of the embryos, and not only looking at, specific features that were selected by the embryologists. The deep learning model can also simplify the grading task, because often different labs, use very complicated grading schemes. And as I said, there is no universal grading system. So it is important, like for example, in our center in Weill Cornell, embryologists use many different grades, like more than 170 different categories for these grades. The AI model that we developed simplify this grading system by mainly, assigning to, like good versus poor quality groups. And this is what we use to train our model. So this is like a classification of good versus poor embryos. So we obtained, time-lapse images of more than 10,000 embryos for our first study. This is more than 50,000 images now. And we used, about 12,000 images for training. We left out a group in the middle. This is like a fair quality group. This is a group that embryologists did not really agree with each other. So we used 6,000 images from the good quality embryos and 6,000 images for poor quality embryos to do the initial training of our model. And then we applied our trained model to blind test sets. These are test sets that were not used in the training. So these are completely independent samples from several different centers. One set was from our own center at Weill Cornell Medicine in New York. Another data set was coming from University of Valencia, IVI Valencia, and another data set came from the UK. So we had a few different external data sets to essentially validate our model. And as you can see on this figure, we observed a very high accuracy of more than 97% for this good versus poor quality classification when we tested on our blind test set. And even like we get pretty good accuracy when we tested on external data set, more than 70%. Why this is very encouraging, This also shows us, like for AI models, we need to, if we want to get, like super high accuracy, like more than 90%, for example, we need to have like a diverse set of data sets in the training phase coming from a lot of different centers. So this is something that we need to address in future, that we should also use, like a diverse set from different clinics. Maybe they have like different protocols that they use, but we should also include that in the training data set. So this is, we can see our paper for more detail, but these are also some figures, like showing the model we use. We use an inception model for deep learning parts. And these are also, like rod curves showing, like a very high accuracy AUC of these deep learning models. So remember, I described a fair quality group. This was a group of embryos that was hard to assess. And essentially, when we applied our model to these groups and asked the model to classify them to poor versus good quality, for the most part, the embryos that were classified as good were those that had a higher likelihood of positive live births. So the positive live births was significantly higher when we looked at prospective data. When we use, like good quality embryos coming from the predictions of our algorithm. So this is also a very encouraging result. So on a blind test data set that we had, we checked how embryology experts grade those images. And as you can see on this sort of like Venn diagram, when we had like more than 200 samples, on only 89 of them, embryologists fully agreed. But the encouraging part was that the algorithm could, for the most part, more than 90% would agree with the majority vote of the embryologists. So this makes us, like get one step closer to the to removing the bias coming from like a wide range of embryologists, because the algorithm can be closer to the consensus. We can essentially use this model, use the predictions of the model, together with some clinical features. For example, the age, which is a very important factor, to basically develop decision-based tree models to predict chance of live births accurately. So we can essentially integrate, like the quality of embryos coming from STORK, our model, together with some clinical features such as age, and use decision tree models to estimate the probability of success in each age group. In order to be, user-friendly and useful in clinics, all these algorithms essentially need to be, we need to have, for example, cloud-based systems or some sort of like good user interface, so people can actually easily upload images and get results very quickly in an easy way. So we developed this user interface for STORK. It's very easy to use. Basically, all you need to do is to upload images to the system, and very quickly you can get the classification results. So basically, you get the probability of good versus poor in this case. And in a more recent work, we can also assess the ploidy, meaning like the euploid versus aneuploid classification. So as I said, the PGTA testing is invasive, and AI models can help predict aneuploid embryos in a non-invasive way, using only image data or some of the clinical information that we have from each patient. So this is a new design that our group did. We again use a large data set for training, more than 10,000 data sets. And as you can see, the breakdown of euploid versus aneuploid data sets here. So we had more than around 6,000 aneuploid embryos, more than 4,000 euploid embryos, and the aneuploid embryos were further classified into simple aneuploid, where only like there's only one aneuploid chromosome, for example, like one type of aneuploidy, or complex aneuploid, where there are like multiple aberrations or multiple chromosomes had aneuploidy. So in order to train our model, we used the embryo images at 110 hours. We also used some additional clinical features, for example, the maternal age, which is a very important factor. We can also use, if we have it, we can also use the morphokinetic time points if a lab uses the time-lapse machines, or if there are assessments of morphological features of the embryos already from embryologists, we can also incorporate that in our model. So we collected this data from our own center, but again, we also use external validation from different centers or different scopes. If there is, for example, a newer version of EmbryoScope Plus was used for additional validation, or you can use like different photography machines and data from those machines to do validation. So this is a model design we had. We used again, we used a convolutional neural network. This time we use ResNet 18, but we added a normalized vector of clinical information together with at the fully connected layer part of the neural network. And this made us do the predictions for aneuploid versus euploid categories. But you can also have a wide range of different classification tasks. For example, you can further classify aneuploid, simple aneuploidy versus complex aneuploidy, or you can do euploid versus complex aneuploid. But as I said, the important feature of this model is that you can also integrate additional clinical features with the model. So I'm showing you one quick result here. And as I said, we used blind test set from our center, but also additional independent test sets from a different scope. This time we use EmbryoScope Plus, which is a more advanced version of EmbryoScope, but all the training data was coming from EmbryoScope. And we also use an external data set from IVI Valencia. And as you can see, even for the aneuploid versus euploid classification, which is the most difficult classification task here, the AI model can achieve close to 70% accuracy for all of these data sets, and an AUC of more than 70%. So this is, again, very encouraging. And when we looked at, easier classification tasks, for example, complex aneuploid versus euploid complex aneuploid versus euploid, we get even to close to 80% classification accuracy. So although this data is early, and this is still not a recipe to basically replace PGTA completely, but you can use it to inform labs which embryos are good candidates to be sent for PGTA testing. So you don't have to do a costly invasive procedure on all the embryos. So just to conclude, I showed you some examples that AI can be helpful and in the field of IVF. Obviously, there's more to be done. We need to assemble diverse sets of data coming from different clinics with variations in different protocols, different populations, for example, to have like a more comprehensive trained model. But as I said, for the most part, we can show that AI models can be very helpful in this field. So just to conclude, I want to acknowledge several teams that worked with my group. First, I want to acknowledge my students, Hosua Barnes and my postdoc, Pegah Khosravi, but also groups from Weill Cornell, the UK, and Spain, who helped us with the data generation and interpreting the data sets.
Great. Are we live now? Is Iman with us?
Yes, I'm here. Hi.
Hey, great. That was excellent. Thank you so much.
Thank you. How are you doing?
Yeah, well, we really appreciate your input. And to the listening group at large, I'll tell you this personally. For me, your manuscripts and your methodologies are very informative. And thanks for your contributions. I did have a couple of questions, and then we can open it up for any chats. I'm going to take two questions, and I'll take the position of a private practitioner in a small clinic somewhere in the United States. And I don't really want to spend money on time lapse. I have a small clinic, but I kind of like this idea of using image analytics to better select embryos. So how applicable will your data set and your image analytics be to my lab, where I'm just taking a single isolated image, but I really want to do this? What's your advice to me?
That's a great question. So actually, some of the models that we developed, they don't require time-lapse data. So we show recently that time-lapse data, if you input the whole videos, would be more predictive. So you can get a bit more accuracy when you want to do these prediction tasks. But you can still do it with still images. That's fine. You can still have only a single image as input for these models and get a prediction. The thing is, if the images from a different scope, we haven't really tested because we didn't have examples of those images in our training data set. But I think we can still retrain models even with images coming from different scopes.
Excellent. Second question. One of the reasons we do PGT is to get as much assurance as possible that we're transferring a euploid embryo, particularly for patients who come to us and say, I want to do everything I can to avoid a miscarriage or possibly even to avoid an elective termination. So the 70% accuracy, I mean, it's astounding. This is excellent. Full disclosure, I'm not a believer in PGT. We do it, but I'm not one of those card carrying, everyone must have PGT type people. So if my patient wants to be as reassured as possible that we're transferring a euploid embryo, How would how would these analytics apply with 70% accuracy? I can do PGT and get almost take it to the bank result.
That's an excellent question. At least the current models and the current accuracy that we have, I don't think we can replace PGTA. So, but if there are labs that like they don't believe in PGTA, that they don't want to do PGTA, maybe they can use our tools to at least, like get some level of confidence that if an embryo is euploid or not. But if there are, especially, I think like these models are better suited for labs that don't want to do PGTA because, like it's invasive, it's expensive. So this is like a very easy way of evaluating embryos. I think it's not perfect because there are other factors involved, and like we don't have the accuracy level of the PGTA itself, but it can be useful to like with some confidence know like if an embryo is euploid or aneuploid.
So playing off that, earlier you had said that this technology might be used to select those embryos that should be biopsied. And I'm assuming that those where the suspicion level isn't high, those deselected embryos for biopsy are eligible for transfer. Is that a correct interpretation?
Yes.
Great. And typically, by the way, I think that the geodiversity that you have in the database is excellent. This is not from one clinic, but you're multiple countries, multiple clinics. So we have one question from a participant is, how many images are you saying, how many images are required? This is not to you, Iman. This is to the, can you embellish on that? Why don't you activate your speaker?
Hi, Mahan. Great to see you here.
Hi, how are you?
Excellent. So just to follow up on what Gerard was asking before, if I don't have a time lapse in my clinic, you said that you could use images to still to run some kind of an algorithm. So my question is, how many images would you want? Because if I don't have a time lapse, I don't want to disrupt the culture. So I may have, maybe additional one or two, or Gerard can actually share from his their own clinic in Seattle how many times they take out and take images, and these time points are not consistent across, even in the same clinic, across different embryos or different mothers. So how would you address that?
I think that's an excellent question. Sort of like depends which model you use to, do the predictions. So, for example, our models, like use for the training data, we use images at 110 hours from time-lapse machines. So the closest you have, even if you have like a single image, that's fine, but they have to be, like as close as 110 hours. So like you get like the highest accuracy. But if you have like enough examples that you can train like a model at a different hour or at like a few different hours, then you can also use, like images at those time points for your test data, or you can just apply to those images. So what we showed in our study, we haven't published this yet, but we showed, like later images are more informative, like if like during like day 5 images are more informative, they are more predictive compared to like earlier images. Yeah.
And what is the area under the curve if you take a single image on 110 versus the time lapse?
Depends, like, because so this study is not published yet, so I can't and we have like several, I mean, I can give you the numbers, but the numbers may change a little bit. But we showed, and it also depends on the classification task that we are looking at, because we have like more classes here. But we showed, like with time lapse, we can improve another 5% on top.
Okay,
of,
that's
yeah. but it depends also like on the age, because age is also like a very predictive factor here. So like with age, without age, models with age, without age are different. If you look at, complex aneuploid versus euploid, is different. It's higher. The accuracy, it can get even like close to 80% because just it's an easier task.
Yeah, I would predict that in the future we'll start incorporating more and more clinical profiles into the interpretation of the images. Same different profiles, I think, will influence how we ultimately interpret. This is just excellent, and I really appreciate your time, and I'll look forward to seeing you hopefully in Anaheim. But we can end here. Excellent. Thank you so much for your time.
Thank you again. Thank you.
Yeah, it's great.
Bye.
Bye.